is growing in part due to the aging of the human population, and a huge number of people is supposed to be ill of Alzheimer's unless new intervention can modify this tendency. Okay, some of the basic features all of you already know is that this is a progressive decay, and this progressive decay in intellectual abilities are due to brain atrophy, and this brain atrophy is driven by first hypofunction and then death of neurons. There are a couple of characteristic deposits, and this is an important point because many of the trials to understand the disease and to try to intervene in the disease are concentrated in this preclinical period, okay, which is very long. And as for the causes, we don't know the causes. Only in a few cases there are mutations, you also know, and in the rest of cases, the high majority of them, we are talking about risk factors. And these risk factors can be roughly divided into two types, one called non-modifiable risk factors. I put these quotes because non-modifiable and modifiable are not as okay, not always a non-modifiable factor can not be modified and the way around. But as non-modifiable factors, the main ones are the age, a little bit is more prevalent the disease in women than in men, and there is a genetic background that is increasingly known because of the existence of big consortia like that one Pascual mentioned, the HESCO in Spain and other ones in the world, that has discovered along many risk factors. Genetic. How are these genetic factors? How do they work? What do they serve us to? There are many of them described, near 100. It is, we know that the risk associated with each one of these variants is very small, but the aggregation of these small effect variants has two main good things to use them, and one is that we can construct polygenic risk profiles, and the other one, and more important, I think, for basic knowledge, is that they help us to detect pathogenic mechanisms, like the one depicted here, that is a recently reported, and that involves macrophages. I don't find the word now. Okay, eat me, don't eat me. This is, sorry. Okay, yes, autophagy. Okay, several points that compose the mechanism of Alzheimer's that we still don't know, but we get some of the information, many data about the pathogenic mechanism comes from studying this genetic background, so it is important to continue to study the genetics of Alzheimer's and dementia. What about the modifiable risk factors? A lot of them are present during our life. There are different environmental challenges that compose this modifiable risk factor, and the good thing is that it is supposed that if we intervene on these modifiable risk factors, maybe that we can prevent almost 50% of the cases of Alzheimer's, modifying these genetic, these non-genetic modifiable environmental risk factors that accompany us from the early life to the adult life. And these modifiable risk factors are mainly education when we are infants, physical exercise and associated in the mid-life, and connection, a connected life in the late life, when we age 60 and more. Okay, well, and there are some trials telling us that some of these modifiable risk factors can really be modified. But to really intervene in a disease, it is important to know the causes. It is not easy, but it is important. And regarding the pathogenesis of Alzheimer's, the first hypothesis, that was the amyloid cascade hypothesis, and as you can see here, there are many complications in this. There are many pathways involved in the amyloid cascade hypothesis, and let's say that to simplify, that all these pathways are supposed to come from an accumulation of A beta. Okay, but there is complications, if we use the most simple of the hypotheses, which is the amyloid one, the picture is complicated. Okay, and beyond that hypothesis, there are many evidences that, not starting from amyloid, but from other places, that can be many, there are imbalances at the cellular, molecular, system, genome levels, many of them. And if you read here, it is not surprising that for a disease with a complexity of AD, the surprise would be if there is a single linear pathway. So the word complexity is associated to Alzheimer's. And as a consequence, there are a lot of causal hypotheses, not many, not unrelated among them, but there are some causal hypotheses, and let's say all of them has some evidence support, some support, okay? And one of these hypotheses is the infection hypothesis, which is really very old. It was first formulated at the 80s, past century, and then it was, let's say, abandoned when the genetic studies were the protagonist, and now it is on the ground again. Okay, so the infection hypothesis from it states that starting from different microbes, viruses that can be present in the central nervous system, in the peripheral nervous system, or outside the nervous system, can somehow start or contribute to the dementia of Alzheimer's type. And all of these agents have been reported, are being studied, and all of them has some evidence, more or less strong. Okay. One of the first agents to be considered for this hypothesis were herpes simplex and herpes simplex type 1, because it is present in the central nervous system, the genome is found in the central nervous system, it is highly prevalent, and this was the main reasons why herpes simplex 1 was the first to be studied. But after that, all of this has been studied. And in fact, for example, this Porphyromonas gingivalis is getting right now very strong support. Okay? And the important question for me is that they can be in the brain or not. They can be in the nervous system or not. So the relations of these pathogens with the neurodegeneration, something that happens in the brain, may also have different mechanism. There is one thing that is associated almost to all the infections, which is the response, the immune response to the infection. And this is general for all these infection agents. For example, okay, let's see here, immune system shows. Okay, what about the herpes simplex 1? This is the most common form of this infection. It infects the epithelia of the oronasal, okay, epithelial oronasal cells. From these cells, it reaches the trigeminal ganglia, peripheral nervous system, and in this ganglia, it establishes a long life latency. Okay, this can be silent for the whole life, or there can be reactivations that are due to several questions. For example, thermic stress, other infections. Maybe some of you here in the audience have this labialis herpes, and you know how these reactivations occur. Okay, and this is known. It establishes latency in the ganglia, it reactivates, and it goes back to the primary site of infection. Okay, it is believed that the reactivations could also drive the virus to the central nervous system, and in the central nervous system, it can also establish latency. And this last part, as far as I know, is still a hypothesis. It has not been demonstrated. If some of you knows something that I not, please tell me. But no, no problem. But as far as I know, the presence of herpes in the brain is true that the genome is there, but we don't know if it is latent, if it will reactivate. But what the infectious hypothesis state is that it is there and it reactivates, and the reactivations repeatedly in the time are the responsible of the of the amyloid deposit, etc. Alzheimer's pathogenesis. Okay, but this is hypothesis. Okay. These are the first evidences. As I told you, they are in the 80s. Okay, you see the one. Okay, these are first evidences, and the current era, let's say, evidences start here in this seminal manuscript of Dr. Ruth Izaki, who found in this year that to have DNA of herpes simplex in the brain and carry the APOE4 allele of APOE is a strong, very strong risk factor for Alzheimer's, whereas none of these agents separately consider a significant risk. It has been repeated by other investigators, and some investigators have found the same results and others not. What I think it is generally accepted is that to have an APOE4 allele helps virus, not only herpes, but also other agents, to reach the brain. Okay, there is an association between APOE4 and viruses, not generally replicated association between APOE4, virus, and disease. Okay. Well, these are other epidemiological evidences. Okay. Peripheral markers like IgM or IgG of high titer. There are genetic evidences, like, for example, this one, genomic studies that give us the idea that infections by this pathway annotation of genes, for example, that we have to be aware of infections, not specifically herpes. Okay. These are epidemiological evidences. There are another long list of experimental evidences, just a few ones. The genomes of herpes are located in the brain, but are located inside or very near to the amyloid plaques. They are not distributed throughout the brain. There are these other one, and for example, in cell cultures and also in animal models, the virus is able to give rise to the main markers of Alzheimer's. Okay, it is able to. It is not demonstrated it does, but it is able to do this. So a recent idea is that these two markers of A beta, of Alzheimer's, both plaques and phospho-tau, are, as this paper by Tanthi, very recent one, states, AD neuropathology may have evolved as an innate immune response to microbial infection. It is also not demonstrated, but there are data supporting this possibility. Okay. Well, so these evidences and these other ones, these are two big epidemiological studies at the nationwide studies in Taiwan and in Sweden with very big samples, stating that two things. These two big studies found that to have been infected in this, take a look at this. The individuals included in these studies were not demented. They were middle-aged, but non-demented. And they were followed by 10 or so years in both studies. And this is in risk of dementia, incident dementia. And what these studies found was that to have been infected is a risk factor, and that taking antivirals for this infection, mainly acyclovir or something similar to acyclovir, precludes this risk. Okay, the virus is a risk factor. The antivirus is protecting these risk factors. This is dementia, not specifically Alzheimer's, and these are not specifically herpes simplex 1, but herpes, both 1 and 2. Okay, so these evidences gave rise to a couple. I think, and I don't know, I think that today, now there is another one, I don't know, but a couple of clinical studies, one in Sweden, I guess, Sweden or Denmark, something in North Europe, and the other one in the United States. Both have finished. From this one, the European one, the results have not been published, and for this one in the USA, they have been published, and they tell it is the font is very small, but they go to the conclusion that valacyclovir, which is the antiviral, was not efficacious in preserving the cognition in these are weeks, so I think it's 18 months follow-up. Okay, it was not effective. Okay, and maybe there are many possible causes to explain this, but there is one basic one, which is that it is the subjects were symptomatic Alzheimer's, whereas the previous studies were made in with treatment previous to the dementia. It is very simple, very obvious, but it is true. So one of the important things regarding clinical trials, not only for this, and Pascual knows this much better than myself, is that we have to pay very, very close attention to the design of clinical trials, because and in fact there are initiatives to take this okay, to make it real, that necessity to design informative clinical trials. Okay, and just another way of going against herpes are the vaccines against viruses. And for herpes simplex 1 and 2, there are no vaccines. Okay, why? I don't know very well. Maybe because the majority of these infections, and for the majority of people, these infections are either no clinic symptoms or mild, especially for herpes 1. But there are currently there are studies to develop a vaccine. Okay, and this is one, for example, very recent. These researchers are looking for neutralizing antibodies, and to do that, they target this glycoprotein B, which is one of the surface in the surface of the viral capsid, which is the main responsible of the fusion between the viral and cellular membranes, which is the way for entry of the virus. Okay, so they are developing these antibodies to this target, GB. They have found some of them which are very potent for neutralizing the virus. All of this picture is complex, but the idea is they have developed very powerful neutralizing antibodies. And another very nice initiative is takes into account this fact. What we are interested in is not only or not mainly to prevent the acute infection, but to get the virus dormant, latent, because many of the majority of us are already infected, and then it is important to okay, both things are important. It is important to prevent the primary infection, but it is there is here we have also to look for something that precludes the reactivations. And this is an example. This is of another other vaccine under development right now, which is targeting the recurrent disease. And in this case, the researchers use B-specific antibodies, one for GB, the main protein responsible of the fusion, and the other is GD, another glycoprotein, which is more specific, which is helping GB, the actor, the helper. And with antibodies B-specific for these two proteins, they protect from recurrent lesions, but whereas a simple antibody, monospecific antibodies, doesn't protect again recurrent, but B-specific antibodies does. Okay, and this all of this is very recent. They are, let's say, we will see what is the final. Yeah, thank you.
I was thinking about the reactivation. I mean, I don't have herpes, but I know that the reactivation frequency is from one person to the other, and it's related to things like stress, or is there, well, is it known how the factors that are influencing in reactivation? Because probably this is more important than the direct viral influence in the brain, but the reactivation, how it changed the immunology probably is important. No?
I think no. Okay.
So are there
How?
factors?
What? I think there is not very knowledge. A lot of there are some things I don't remember now exactly. Some examples, for example, of what is known. I think this step, latent and reactive action, is very difficult to study. For example, it's very difficult. So I think there are not many data because it is not easy to study. And for example, there are models, animal models of latency and reactivations, but they are animal models. Okay. There is also some very recent an MPC cell model in which the researchers are able to establish latency and reactivates, and but I don't think there is enough knowledge to tell you this is a possible target. They are standing. It is one of this is our main interest right now, actually. But maybe if any other people, I repeat, has some information I missed, please share with me or with us. Okay. But I agree with you. This is a very important point to focus on. In some way, reactivation is not very different from the first infection, because once the genome is activated in the neuron where it was dormant, it has to go to the neighbor cells and transmit from one cell to the other one, and this part of the reactivation may be similar to the first one. And in this sense, for example, here the antibodies are directed toward the entry of the virus, and they get a protective work of the agent, because probably they are able to cut the transmission from cell to cell, not the reactivation by cell in the same cell, but the transmission. So in some way, to get an agent that inhibits cell-to-cell transmission is able for both first, and for example, or another example is as acyclovir is prevents recurrency somehow, not at 100%. So there is something to many things to learn here. How the reactivation and primary infection are similar, are different. So very interesting question, but I don't know the answer. See? So, and this is the table with many columns, many rows. Those are all the family of the members, the major members of the family of herpes viruses. Almost every one of them has been somehow related with neurological manifestations. From these viruses, those three first are alpha herpes viruses, like as a subfamily. All these three establish latency in neurons, whereas other members of the beta or gamma subfamilies also establish latency, but as far as is known, not in neurons, in other cells, in or outside the nervous system. Okay. Well, most probably you have been hear about this varicella zoster virus, because it is thought that this is the only epidemiological demonstration, the researchers told that, of a causal relationship between one virus, one herpes virus, and cognitive decline. Okay, it is called a natural experiment because in Wales there was a time where everyone being born 1 week before that time. Here is the day, the specific day, were not eligible to be vaccinated, they were not vaccinated, and people after that week had the opportunity to be vaccinated. And in fact, more than 50% of those people who were allowed to vaccinate that did actually get the vaccine. Okay. Well, accompanying this step in the time, there was a decrease, significant decrease in the appearance of the lesions. The singles are those lesions associated to varicella zoster, the culebrilla, okay? And also, there was a significant decrease in this manifestation, a very, 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 very bad manifestation of the shingles, which is the neuropathy, post-herpetic neuropathy, and also small but significant decrease in the diagnosis of dementia in a period of several years. Okay. Well, so there are evidences that the infectious hypothesis infectious agents may be related with Alzheimer's, virus may be related with Alzheimer's, demonstrations not by now, and we started to approach this question many years ago, in fact, more than before, more or less, when I know Pascual, when more or less. And the question, we were, by that time, we were also working with neuroblastomas, cell lines, a very simple model, cellular model. And we were trying to answer questions like mutations or oxidative stress, were able to kill the cells. How do they kill the cells? And from these cellular models, we were looking for candidates to interfere with pathogenic mechanisms. And one day in the lab, there was I was working with Fernando Valdivieso at that time, and he came to the lab and he told us, I have seen, it was the paper of Ifsaki. I have read this paper in 1998, and I have an idea. I think that if there is something in the brain, it should be doing something. Okay? If it is there, it must do something. Could you demonstrate me that it is true or false? So we started to try to make the hypothesis of herpes involvement false. Okay? And okay, to do that, we include the infection of our cellular models as the main trigger. And these things here are when we find something in the models, we use this. Okay, we go to the humans, thanks to our also long-lasting collaboration with the neurologists at Hospital La Paz. And after that, we go to the consortium. Okay, well, we look for targets in cells. We try to validate those targets in humans, and this is and that's all. Okay, just in 5 minutes or so, I will try to go to show you a panoramic of what we have in this. Okay, with these experiments, telling a little bit what are we doing with cell models, what we have here, what have we found. There's just a panoramic view, as I told you, biomarkers, and also what are our main collaborations in this project, herpes virus and Alzheimer's in Cybernet. Okay. Okay. We come from experiments in neuroblastoma, human neuroblastoma, that showed us, and it is past, okay, that lysosome and cholesterol metabolisms are prominent consequences of herpes infection in those neuroblastoma cells using transcriptomics and then functional analysis in the same lines, these papers here, okay? So when we moved to novel cell models, what we were trying was to use more realistic models. This is a human neuroblastoma. In particular, this one does not differentiate very well to mature neurons. So it is very artificial, and we wanted to go to a more realistic, let's say, cellular models. And to do that, we selected something that is experimentally not as difficult as induced pluripotent stem cells. It is not easy anyhow, but we started from cellular cell lines, from neuronal stem cells, and these neuronal stem cells came from the midbrain of human midbrain, because we don't want it to study the cortex, because it is where the virus, where it is most probable that the virus enter in contact with the brain from the peripheral nerve is the brainstem, not the cortex. So we selected brainstem as the site where the cells come from, and we selected 2 immortalized NPCs by the reason that this one is differentiates to neurons, and it has been reported that it can establish latent infections with herpes simplex 1. And this other one is able to differentiate to neurons and also glial cells, astrocytes mainly. And here we can then study the role of other non-neuronal cells abundant in the brain, okay? We put in several platforms, 2D, 3D, for the two lines. And then we studied what we always study: death, neurodegeneration markers, and we have not yet go there. Then transcriptomics. We are here. We are still not make a transcriptome study of these cells, but we have some data. What have we been able to go to the reality? To put it in the slide is easy. To get the cells here is not. It's long. It's a long way. No, I don't know if any one of you is working on cell lines, but those 3D cultures are not easy, are not very stable. But we have been able to develop 3D spheroid-like aggregation of both cell lines. We have been able to have them in growth. We have been able to infect them, and we have found that, as was the case in neuroblastoma, also in Lumes and Ren cells. I also showed you here the Ren cells. Cells are able to differentiate in this spheroid-like, let's say. We infect them. We have we have a recombinant green fluorescent virus, which facilitates very much the following of the infection, and the infection is able to induce amyloid and tau deposits. So the virus is so these neurons and also the glial cells are susceptible to the infection. Both of them accumulates amyloid and phosphotau. Okay. What about biomarkers? The humans. Working with a series of cases and controls from the Hospital La Paz, we have been able to show that there is alterations in peripheral cells in cholesterol. There is a decrease in total cholesterol in all the immune cells in blood, and there is an increase of CD60 in innate immune associated cells. Okay, so blood biomarkers support the involvement that does not allow us to discard that cholesterol and immune system are altered in the periphery of Alzheimer's patients. Okay? And this is also what we found in this series. This is a presence of IgM. IgM is the main indicator of active, maybe primary or recurrent disease with virus. And we found this figure, which is, if you remember, similar to those reported by Itzhaki, the convergence. If the two things are true, okay, we have IgM and we have ApoE4, it is there is a risk much higher than any one of them separately. It's more or less the same idea. As you can see, this is a tendency, no? Not significant. We have not get enough. Maybe we have not get enough sample size. Maybe, or maybe this is not true. In any case, it is a tendency. We have also found that, and this is, I think it's interesting because it uses subjects before the onset of dementia. They are healthy people. This is in collaboration with Dr. Cantero of Cibernet, who is in Sevilla. And we use this population. And what we found is summarized here. In these people, which are not demented, they are cognitively controlled, correlates the immune response measured as levels of IgG, specific for herpes simplex, correlates with amyloid accumulation in the brain. And this could be in line with a possible role of amyloidogenesis to against infection before cognitive impairment, in line, let's say, with this proposed role of amyloid as an immune response postulated by Tanzi. This is in line. Okay. Finally, we have started to study also the role of herpes with another cells abundant in the brain. In this case, oligodendrocytes. We have started almost. We have 2 years or so of this project. This is the foundation for this collaborative project. Is that Dr. Cantero found that there is a correlation between herpes and amyloid. In other work, he found that there is a correlation between brain myelin and amyloid, and the question is, there is also a relation between these two parts. This is demonstrated. This is demonstrated. What about that? Does herpes has something to do with myelination in the brain? And to start this project, we are very lucky because we have these expert collaborators in the project. Eva Carro, who is the coordinator, has a very big knowledge on lactoferrin, which we add also to the project to try also in this project we try to find the relation between herpes, myelination, and if there is a protective role of lactoferrin. This is the whole project, let's say. We have this component which has the necessary knowledge for each of the parts. And we use mice to in vivo and in vitro, study the relations between the virus and the oligodendrocytes, the myelination part. And we also have humans, two kind of human things, brain tissue from mainly from here, from the Fundación, to look in those samples, herpes, myelin, and A beta, to try to find them together. Or and we also have studies in vivo, studying the relation between herpes in blood, as we did in the previous work, and myelin followed in the brain. Okay, this is the whole project. We have some data, and the most advanced comes from the in vitro studies. We are starting in vivo studies in mice, but in vitro we already know that the virus is able to infect oligodendrocyte, as well as it was able to infect neurons, astrocytes. Also, oligodendrocytes are susceptible, and they also accumulate A beta and phospho-tau. We have I have only the A beta part, but they also accumulate phospho-tau, and lactoferrin is very potent, protecting from the infection. In green we have the virus, so there is no virus, and there is no, in red, A beta and tau accumulation. And we are working on publishing, to publish it. We have not yet published that. Okay. So just to try to summarize, this is something similar to my brain when I think in herpes. This is I think it is nice to read this review, which covers the relation of many viruses with the brain. There are many viruses, there are many cells, and almost all of the relations you can think of are possible. So working in this complex scenario and starting, what have we been able to demonstrate if the virus is or not related to Alzheimer's? Well, this is right now we have not been able to discard it, which was the first intention 25 years ago. So the evidence doesn't allow us to discard the role, but of course we have not been able to prove the causal intervention, and this is my belief. I think the group is also in this line, so I put our, not mine, but I think the evidence support to keep working on this, and I hope to have convinced you that it is worthy. Do you think so, or not? Okay, well, thanks to you for the patience with my English, and the group, the clinic group, all the collaborators, both researchers and patients, that allows us to continue working on this. Okay. Question?
Questions?
Oh, thank you, María. Well, my belief, I am convinced about this. I mean, very much, very much. And we, we worked within FUPE in the past. And actually, I think there is a pattern. You look for specific microorganism related to Alzheimer's. You find it related to the lesions in tissue. You find it in CSF. You find it in high vulnerable regions, better than in low vulnerable regions. I mean, there is a pattern that has been reproduced in several viruses, particularly herpes simplex, but also in what you say with Porphyromonas, the treponema, and the fungi. What we did in fungi, we found. So actually, I am quite convinced of the polymicrobial hypothesis. I mean, there is not a single, probably, I mean, probably one single agent, infectious agent related to Alzheimer's and other neurodegenerative diseases, but polymicrobial, which is a very, very, very difficult thing to study, particularly from an epidemiological point of view. So yeah, this is what do you think about that?
Oof. I agree almost with almost everything you have said. I am convinced. I am convinced that there is no one microbe. I am convinced that it is not easy. I don't know how to. Okay, I agree with you. And okay, I think there are some things we can think of, like I don't know how much of the evidence can these things add, but the study of the virome, for example, has not yet been performed many times. So I think it can give us some answer. Maybe it can, if the genome can answers can learn no, the other way, teach us can teach us about genome. I think, for example, virome could be somehow useful. No, I have no more ideas, really, because I agree with you that there is no one agent, but there is also there is a difficult question to answer, which is the classical one: what was first, the agent or the leakage of barriers?
Very nice talk. Pretty convincing. Your English was very good. First, I was particularly impressed already in the first slides with this difference between the APOE positive and HSV1 positive versus negative. And this, I think, odd falls of 30.
Yeah, in that,
30.
yeah.
So it seems that maybe when we see the effect of APOE in the global population, we are seeing the interaction of with the virus, because actually in the two things, the risk when you are not positive of being only APOE positive was not that bad. So maybe this 8, 10, 12 fold that we see in the general population is because we are compounding these 35 folds with people that actually is positive but doesn't have a low risk. So maybe would it make sense to include this interaction as a risk factor for re-stratification, maybe for clinical trials, instead of only APOE, I mean?
Well, yeah, I have something to tell you about that. Is that one thing is that, well, as when we study APOE risk, of course, we are including other agents, and our herpes is one. But what is the power, the weight of the infection is difficult to answer, because we don't know how many people has APOE in the brain. To that interaction was between APOE, easy, the herpes simplex genome in the brain, not easy. And in fact, when other people has tried to replicate this, not all of the researchers have found the same so powerful interaction. The interaction between APOE and indirect signs of the infection, for example, serology in blood, would be interesting. I think it would be interesting, but because it is here also. Also, I don't. There are some studies, but not so much to can calculate the interaction factor between ApoE and serology in blood. But I think, of course, it is. I think it's interesting, of course, to study serology in blood. It's interesting and affordable. Not as not.
Because I wonder that if it's tested in blood should be very affordable too.
Yeah.
And another question, maybe not for you, but probably to for Dr. Cantero more appropriate, but in this association that you found with the amyloid deposition, these areas are not the ones that you would expect amyloid to be deposited in the brain of people with Alzheimer's. So I wondered, probably I will read the paper when we go out here, but which is the explanation or why these associations in these strange areas are not in the areas of the brain that would make more sense, I would say. And also the associations that survived in the, I think the rostral anterior cingulate, these areas are very well known that deposition of amyloid here cannot be captured by PET. This has been shown by many pathological validation studies. So I wonder, this is the discussion.
Yeah, you're right. I'm not an expert in neuroimage of the brain. Just a little thing is that they are not Alzheimer's. They are not preclinical Alzheimer's. They are amyloid negative. Yes, they are somehow at risk because of they have some familiar, but they are cognitively normal and they had no amyloid in CSF. They are not preclinical Alzheimer's. They are healthy, cognitively healthy. So? Okay, yeah. Yeah, sorry, but really you are right, and that why these regions, why not other regions? It's Dr. Cantero. Sorry.
So I have a specific question that brings me to a more general discussion question. The specific question is, I was intrigued by the clinical trial that you show, the one that was negative.
Yeah.
And then, of course, it's negative, then it was too late, but it was not only negative. It appeared.
Yeah. You have seen that number, eh?
So that's kind of not the same as negative.
I don't know if I can go back because I don't know if there's other one. Aquí, here, mejor. But you are right. Not it is that figure is not only it's the other way around. So it is bad for cognition to have the.
So what happened there?
I don't know. That result is very recent. I don't know. They don't explain in the paper why. And I'm waiting for to answer you. I'm eagerly waiting for the other clinical trials. But you're right. There is a significant worsening in the people treated with valacyclovir. I have been studying because I read the paper. I didn't see the answer. I was looking for doses of times, and maybe that dose for so long time, which is 1 year and a half, 18 months is too long. I don't know, because it is supposed to be very safe, the valacyclovir, but I'm not completely convinced yet. I have not had the time to study in deep, but it has something to. Did you see this? Huh? You saw this. You were very. You have a good side, and yeah, it is true.
So that
The-
makes me bring me to the more general, almost philosophical question,
Yeah.
which is, of course, in a situation with so many recognized risk factors, does it really make sense to try to tackle one by one, risk factor by risk factor, with a it's sort of kind of assumption that if it is a reductionist assumption, then if we tackle one by one, we can kind of solve the problem overall. It could very well be possible that you try to tackle one and mess up by
Yeah.
resolving one, you start mess upping with the other one. So all the interactions between all these mechanisms become the key. So it's a sort of labyrinth we will never be able to get out of going one by one. What do you think? And we might have to think about something that is maybe some more general type of approach, like sleeping more. That is, we can kind of get bridge a lot of those mechanisms. Or what are the interventions that are more bridges rather than mechanistic? What is your take in this problematic?
Well, I think there are some things that are more generic and then can be more worthy to try to target. For example, but the things that are generic can protect from many different entries. For example, I would say it seems that inflammation is not a good thing. For example. And when I started to study Alzheimer's, in fact, before Itzhaki, before genes, there were some big clinical trials by then showing that IL-6, IL-6 was very, very, very bad, and it was. I don't remember very well what was the clinical trials, but I remember that were impressed in that time by the strength of that IL-6 cytokine. And this is a mediator of immune response somehow, and the immune response is also very complex. But when that IL-6 were to clinical trials with anti-inflammatory, the clinical trials failed. But I'm convinced that, for example, high levels of inflammation are not a good thing for the body, not for the brain. How to tackle that? But no. Ya yo había terminado ya. But
No, the thing, I was actually my comment, my previous comment was about that. Yeah, about the polymicrobian. But the thing, I mean, there is something like a paradox in maybe has something to do the way research is done and presented. But each agent, each infectious agent that is famous in Alzheimer, herpes simplex, zoster, treponema, porphyromonas, and there are quite chlamydia. Everyone has a very, very powerful evidence behind, very powerful. I mean, the evidence on zoster is, I mean, it's evident. There is something to do with the evidence you present on herpes simplex is overwhelming. The evidence on treponema is, I mean, you look on senile plaques and they have treponema DNA inside. So therefore, it has to be something very complex, very combination of. But I understand that people try with clinical trials try to treat specifically one infection. I mean, porphyromonas, that paper some years ago, porphyromonas, that was in Science Advance, I think. It was, I mean, you find the protein that porphyromonas secretes is inside neurons with tau, and only neurons with tau, with phospho-tau, say, that's it, we found it, that's the cause. And that is always with infectious. I mean, that there is, therefore, I think obviously there is an influence, has something to do, Alzheimer's with infectious agents, but with many infectious, maybe, yeah, it is at another level. It is at the inflammatory level, immunologic level, or so. But I understand that people go and try to treat, and maybe, yeah, that polytherapies could be, or clinical trials trying to address different agents could be somewhat successful, yeah.
I think yes, because you were.
So I wonder, maybe it's for you, how much is possible retrocausality type of effect, so that there is something that makes vulnerable to Alzheimer and vulnerable to infections. So at the end, you tend to have, when you're at the end on the pathological level, the two tend to concur. And so at the end, we cannot, even a poly type of treatment would not work because there is something ahead that makes the brain vulnerable to both, or something like that. So when you say there is a very strong evidence, how strong is that in the direction of the causality?
Oh, causality. That's
has to do with a
causality is the word.
with no, I mean, now yes, but some years ago with no
Está bien.
effective therapy, and the way we present the data, we published in scientific reports, we published with Luis Carrasco, the presence of different fungi in different regions of brains, Alzheimer's, not Alzheimer's, and it was a very powerful evidence. I mean, all these fungi had something to do with Alzheimer's. This paper has now hundreds of citations, but nobody. I mean, there is no evidence that anti-fungi agents are have any effect on Alzheimer's. So I think that has something to do with the way we interpret our results as causal, as you suggest. Yeah.
We have.
Yeah. Just.
Okay, a brief comment and then a brief question related to the study that Jesus told before about the amyloid beta and the herpes. Maybe my hypothesis will be that if it's related to myelination, or for example, the herpes in the oligodendrocytes, these regions are very, there is a high plasticity in those regions, and maybe one of the reasons could be that the oligodendrocytes, if they have herpes virus, and as these regions are more actively myelinated and demyelinated because of the plasticity, maybe this could be a reason why they are more vulnerable to that. Because in other regions that are more myelinated, they are more stable in the myelination. It could be a possibility.
Yeah.
And then my question was like, if you can, I mean, I'm very curious about the relationship or the processes that are behind the connection between the herpes and the cholesterol, because of course, and the relationship with ApoE, because of course ApoE, one of the main roles of ApoE is to transport cholesterol.
Yeah.
And cholesterol is also very necessary for myelination.
Yeah.
But I don't understand, or I don't know, or maybe it's not known the relationship between herpes or the effect of herpes in cholesterol. I don't know if you can give us more information about that.
I can tell you some pieces of
Okay.
virus. This virus, herpes simplex, has a membrane, so it needs, and it's proven, it needs cholesterol to form their membranes. There is one of the relation is that one. More relations. The virus activates the endolysosomal pathway and then stop it. Like the virus makes this same action and then stop to apoptosis, to different kinds of cell death, and also to the lysosome. It increase the cascade and then cut at the lysosome level at the end. And in this pathway, cholesterol is involved in the lysosome membranes. It has been it alters cholesterol traffic in those medium final vesicles. I didn't find the I didn't find the word vesicles. What's the word? It alters the traffic because many of the cholesterol traffic goes through vesicles, and there is this is other interrelationship. We have found in the neuroblastoma that the virus induced high levels of cholesterol and also alters that traffic. It is an experimental data. And we have also found that decreasing with methyl beta cyclodextrin interferes the infection. So there are several evidences. I don't know, maybe I forgot some of them, and I don't know if this makes a complete picture, but pieces, like of everything else.
This is definitely a very interesting topic, but I think we have to stop here.
Yeah.
Maybe we will invite you another day to continue the
Okay,
discussion. But
if you are able to follow me in English, I can come back.
thank you very
Okay.
much. Thank you.
Thanks to you for the interest. Thank you. Thanks very much to you for the interest. Yeah. Qué bonito tu pregunta esa. Es que eso sí que no, sí, es que sí.